Well, if we can't fix the kidney, we can replace it. And that's what, of course, transplantation is all about. And our next speaker, Dr. Manikkam Suthanthiran, is Stanton Griffiths Distinguished Professor of Medicine here at Weill Cornell and Director of the Immunogenetics and Transplantation Center at the Rogosin Institute. He works broadly in immunology, not just in the kidney, but in liver allograft rejection. And he's going to take us through some of the immunological challenges. We need to be able to make transplanted kidneys survive longer and serve better because the supply is limited, because Leif has just told us he won't be rebuilding them for a little while. But we'll get there eventually. So Transplant Suthanthiran. Suthan.
Thank you, Barry, for the introduction, and I'm delighted to be here. It's been a great meeting. Before I give my presentation, I'm fully aware that I'm the only person standing between you and your lunch, so I'll make sure you're out on time. Transplantation has benefited remarkably from advances in immunology and molecular biology, and in the next few minutes, I'd like to share with you that data. Okay, I need to know how to move this. Okay. On December 23rd, 1954, an historic event took place at the then Peter Ben Brigham Hospital. This was the first successful organ transplantation. It happened to be a kidney transplant between these two individuals. And as you might see from this slide, they actually happened to be monozygotic identical twins, so there was no immunologic barrier to breach, but this remarkable accomplishment was possible by some of the pioneers shown in this slide also. This is what happens when you give an immunology talk. Someone always turns on their beeper or their cell phone. Here is Dr. Harrison. He was the surgeon who did the donor nephrectomy. This is such a common procedure in year 2010, but this was the first time in the history of medicine a perfectly normal person was taken to the OR and an organ was removed. A major procedure was done with no direct benefit to that particular patient. And there was considerable debate at this time in 1954 about doing this. This is Dr. John P. Merrill. He was the first example of perhaps a transplant nephrologist under whom many of us had the privilege to train. And this is Dr. Joe Murray, the transplant surgeon, who not only devised this surgical procedure, but also made several innovations that made transplantation possible. This is in 1954. 50 years later, here is Dr. Joe Murray. 2 years back, I had the privilege of being the Paul Russell Professor at MGH Harvard. And Joe Murray was there, and many people who have made major contributions to the transplantation field were there. And it was a great opportunity to review the progress, the challenges, the promises that were kept, and the hopes to be met. And this is what I'm going to review with you. In 1963, there was the first World Conference about transplantation experience. About 200 transplants or so had been performed. And if you can look at the survival rate of patients in 1963. In fact, I asked Barry before earlier in the morning after his wonderful talk what he thought would be the survival of patients in 1963, and he's working for CMS, so I don't expect him to be too optimistic, and he said it probably about 30%. But as you see in this slide, it's actually 10% for the living donor, and not a single recipient of a deceased donor kidney survived in 1963. Where are we now? Here is the survival rate published by the United Network for Organ Sharing. It also tells you the kind of transplant activity in the United States. The kidney is the most common organ. We do more than 10,000 transplants a year. The second most common is liver, then heart, lung, kidney, pancreas, and pancreas alone. And look at the survival rate. In the relatively short span of 50 years, transplantation has moved from a highly experimental, high-risk procedure to a safe therapy, with survival in excess of 80 to 85% across United States. And this is, we talked about electronic record keeping. This is a well-documented information because each transplant center has to report their data back to the central database. And some of the reasons for this spectacular progress in our field: one is an improved knowledge in immunology and molecular biology. We are fortunate recipients of several outstanding drugs that control the immune response, the infectious prophylaxis, because our patients receiving all these immunosuppressive drugs are ever at threat for twin complications of infections, life-threatening infections, and we have very good drugs to manage them, both viral and bacterial. I know this is an eclectic audience. You are not card-carrying immunologists, but nevertheless, I'll have to take you through some of the immune responses, and I basically follow this principle in showing my slides. This used to be one of the slides I used to start my talks with, and when my first slide was this, people did exactly what one of our physicians is doing, paged themselves out of the room. But I think it's very important we kind of take you through this because it captures, it's really not very complex when you'll see. So when we want to do a transplantation, we follow, the first principle we follow is the principle of transfusion. If you can do a transfusion between two individuals, you can share the organ between those two. That's the standard procedure. But we are capable today, in 2010, of breaching even the blood group barrier. But in a conventional, standard transplantation, if you can do transfusion, you can do transplantation. The second most important system is called the HLA antigens. The HLA are human leukocyte antigens. Everybody in this room will express these antigens on your cell. This is how you distinguish self from non-self. This is the antigen system you use to protect yourself. When you think about this, nature could not have anticipated transplantation. So these antigens were not placed by nature to frustrate us. These antigens are there, so when an organism, an infection comes in, these HLA antigens can bring them and present them to our immune cells, and you can mount an immune response. The HLA class 1 is expressed on all nucleated cells. Class 2 have a much more restricted tissue distribution. These are expressed on kidney allograft and every other type of an allograft. And this is the donor kidney. These antigens are seen by the recipient T cells, which can be broadly classified into CD4 T cells and CD8 T cells. On the surface of these T cells, there are multiple receptors. If there is something to be seen, you need appropriate receptors on these cells. And these receptors include what is known as an antigen recognition complex that induces CD3 protein. It's a gamma, delta, epsilon, and 2 zeta chain that's in linkage with an alpha beta T cell receptor. Together, they transduce the foreignness of the transplanted organ into the cell. And this signal transduction requires a number of other molecules that are shown here. But the net effect is the recipient is able to see that this kidney is a foreign kidney and make a variety of growth factors called cytokines, acquire receptors for these growth factors, and this interaction between the growth factor and the receptors results in the differentiation and acquisition of specialized function. In many ways, the way to think about cytokines is that they are like the language. This is the language the cells used to communicate with one another. So this T cell is a naive T cell. Now this T cell now is now well educated. It has is basically gone through a program that is genetically determined. It's well orchestrated, and if unless blocked by immunosuppressive drugs, will destroy the transplanted organ. We are very fortunate that we have there have been a number of new drugs. The first drug that was used in clinical transplantation was azathioprine and then corticosteroids. But in the early 1980s, a new drug was discovered called cyclosporine. This is a product of a fungus called Tolypocladium inflatum. This came from the soil fungus. John Borrell was the discoverer of cyclosporine's immunosuppressive property. We understand how this drug works to some extent. Here is the cyclosporine. This binds to a protein called cyclophilin, which is a peptidyl prolyl cystrans isomerase. Every one of you, in every one of your cells, you express this protein in large abundance. This binding of cyclosporine to cyclophilin now results in cyclosporine acquiring immunosuppressive activity. It goes and targets an enzyme called serine-threonine phosphatase calcineurin. And calcineurin is important for the generation of transcription factors such as NFAT or dephosphorylation of this transcription factor, which is very important for the expression of a number of genes. And this next slide shows an experiment we did in the early 90s. The details are not important. This is a transcription factor, nuclear factor for activated cells. This is only expressed in stimulated T cells, and this is blocked by cyclosporine. When you block this transcription factor, you do not express the IL-2 gene. Here is a stimulated T cells expressing the gene. Here is a T cell with cyclosporine that fails to express the gene. This IL-2 is also known as T cell growth factor, and you need this growth factor to expand the T cells, for the T cells to divide. And so when you block this growth factor, the T cells don't divide, and you're able to prevent rejection most of the circumstances. A very similar drug is called Tacrolimus. This is also a natural product. It was introduced in the clinic by Tom Starzl and another true pioneer in the field of transplantation. He basically invented liver transplant, and tacrolimus in many ways works in the same fashion as cyclosporine. So in clinical practice, we use either cyclosporine or tacrolimus, and in the United States and outside the United States, it's very likely 90% of our transplant recipients, be they be recipients of the heart, liver, kidney, would receive either cyclosporine or tacrolimus as their treatment regimen. Suren Sehgal discovered this drug called rapamycin. Rapamycin in a fashion similar to tacrolimus binds this particular protein called FKBP12. This protein is also a peptidyl prolyl cystrans isomerase. This complex of RAPA and FKBP2, instead of targeting calcineurin, targets an enzyme called mammalian target of rapamycin and blocks cell cycle progression. One of the most fascinating things about rapamycin, it's not only a good immunosuppressant, it is able to prevent the proliferation of immune cells as well as non-immune cells, including fibroblasts, including malignant cells. So there is an opportunity here with a drug like rapamycin that you can not only block the immune response, but you also have an opportunity perhaps to block fibrosis. And very importantly, in experiments done in a laboratory, including ours, there is actually an anti-cancer effect associated with rapamycin. And transplant patients are always under threat for the development of malignancy. So this rapamycin now provides a unique opportunity to buy immunosuppression, but also have an anti-cancer effect. This was demonstrated actually by one of our renal fellows, Fu-Lung Lan, who is now at the University of Michigan. And 2 years back, or last year, the FDA approved rapamycin for the treatment of renal cell cancer. There are a few other drugs. I'm not going to go through in detail the exact the biochemistry of it, but a drug called mycophenolate mofetil, which is a much more a refined version of azathioprine. I see Dr. Ray Sherman just walking out as soon as I pulled up this particular slide. Mycophenolate is an inhibitor of nucleic acid synthesis. It inactivates an enzyme called inosine monophosphate dehydrogenase, so it's a very good anti-proliferative agent. In addition to these drugs, rapamycin, tacrolimus, cyclosporine, and MMF, we also have access to a number of antibodies. One is called anti-CD3 or OKT3. This was the first monoclonal antibody ever used in human being. It targets the gamma chain or epsilon chain of the CD3. Since CD3 is expressed on all post-thymic T cells, you have an opportunity to eliminate T lymphocytes. T cells are absolutely essential for a rejection of a transplant. So if you knock down T cells, you can get a very effective immunosuppressant. But this is a mouse monoclonal. But when you give a mouse monoclonal antibody to a human being, you can recognize the antibody as foreign. So through genetic engineering, the monoclonal antibodies have been humanized. That is, the antigen binding portion of an antibody is retained. The rest of the architecture, in like the FCPs, it comes from a human immunoglobulin. So this type of a humanized antibody is very effective and doesn't cause the HAMA response, human anti-mouse antibody response. The other agent is a polyclonal agent called thymoglobulin. We use it quite a bit in our program. This kind of reacts with many cell surface proteins, thus functioning as an effective induction agent. So if you take all this together, one can think of how we apply these principles to optimize transplant outcome. The first thing we do in our program in the United States in renal programs is to match the recipient and the donor for HLA class 1 and class 2 molecules to the extent possible. Every patient in the United States who is waiting for a kidney transplant is in a single list. So there are about 70 to 80,000 patients today waiting for a kidney transplant. They are all registered in a central database. When an organ becomes available, the tissue typing laboratory will type that donor and put that information into the central database. US Congress actually passed a law about 20 years back mandating that if there is someone who is perfectly matched for a kidney, that kidney should be shipped. Let's say a kidney comes from a New York hospital. We harvest the kidney from a deceased donor, and that kidney happens to be matched to someone in California. By US law, we have to ship the kidney. So in clinical practice, we try to match as much as possible. But this antigen system is the most complex antigen system known to mankind. In HLA system, there's an A, B, C locus. Each locus has 50 antigens. In class 2, they are DR, DP, DQ. Each of them have 50 alleles. So if you like shuffling the cards, if you look at the permutation combinations, the matching is very difficult. Fortunately for us, there is not a random allocation of the alleles because during the evolution, certain combination has been favorable for our survival. So there is an event called linkage disequilibrium, so certain antigen travel with certain other antigens. So if we do 10,000 kidney transplants, we are able to match about at least 300 to 400 of them perfectly matched. But then we go by different levels of matching and we give some scoring system. We use multiple drugs. These drugs are wonderful, and every drug has a toxic side effect, but we like to use like corticosteroids to block IL-6, cytokine, monokine production, cyclosporine, FK506 steroids to block IL-2 and interferon gamma, azathioprine and rapamycin and MMF to block clonal expansion. In a way, it's like almost like a smorgasbord approach in immunosuppression. You take one from one class like an appetizer and an hors d'oeuvres or a main dish and a dessert and a coffee. You kind of choose different agents that work at different points of the immune system in order to maximize the efficacy and minimize the toxicity. In our own program here, it has actually a very long and distinguished history. The transplant program was started in 1963, and actually I see Dr. Kurt Stenzel in the audience. Dr. Al Rubin was shown earlier with the dialysis mission. Dr. Stenzel and Dr. Rubin participated in this pioneering event. This was first in the tri-state area. As of year 2007, we have completed 3,000 kidney transplants. We now do about 200 plus transplants a year, and almost half of them come from a living donor, and half of them come from a deceased donor. The survival rates are publicly available, so patients can look at the data and see which center they should go to. And our survival rate, in the last published data in January of 2010, was a 97% at 1 year and 95% at 3 years. Graft survival rate was 94% at 1 year and 87% at 3 years. And actually, some of the survival rates are statistically higher than what one would get for our case mix. This is because we have truly an interdisciplinary team managing this patient. And this is not just physicians and surgeons and immunologists alone. It requires the pre-transplant coordination, nursing personnel, the whole group of individuals. And this is to me, one of the most rewarding aspects of being involved in transplantation. People really come together when you make rounds on the floor and you see an individual who just got a kidney this morning, you can't imagine how many people participated in that organ reaching that particular person, not to speak of the wonderful generosity of someone donating an organ at a moment of extraordinary stress, and also about the living donor. In many ways, the organ donors are the real heroes of this story. We have several problems still in our system. It's not, even though we have a 90%, 95% survival rate, on a day-to-day basis, when you take care of this patient, in a single patient, we have several challenges, and I'm just going to touch on a few. Here is a kidney, normal kidney. Here is one called acute rejection. This is the organ is suddenly seen more immunogenic by the recipient, and the recipient mounts an acute immune response resulting in graft dysfunction. Everything is going along fine, and suddenly the graft function goes down, and this is acute rejection. And there is also a slowly progressive process called chronic rejection. And this is a major challenge in transplantation. And we and others have been studying this process. And our own studies have benefited remarkably by this wonderful invention by Kary Mullis, for which he received the Nobel Prize in 1993. This is called the polymerase chain reaction, where you can actually take one copy of the gene and in a test tube multiply it into about 210 million copies of the gene by simply doing what is known as a PCR cycles, designing the primers appropriate to the gene of interest. And this is, people say this is like picking a needle from a haystack. It's actually picking a needle from a needle stack. From a 20,000 or 30,000 genes in a single test tube, you can pick one particular gene you want and make millions of copies of this gene. When Kary Mullis described this protocol, he actually described a qualitative PCR. Bagui Lee in my laboratory in the early 90s made it into what is known as a competitive PCR or a quantitative PCR. So you can actually put a number on the genes you are looking at. You can say, if you want to say BMP7, one can say you got about 5,422 copies of BMP7. So you can actually quantify the copies. We did this. We actually were able to show that in a transplant patient, clinical patient, if you measure in their urine from a renal transplant patient, certain genes that participate in cytotoxic T cell activity, and these genes are called perforin and granzyme B, if you measure those genes in the urine of transplant patient, you can actually predict the status of the transplant with respect to presence or absence of rejection. In fact, we studied BMP7 during acute rejection, and we find BMP7 to be decreased during an episode of acute rejection, consistent with an idea an acute rejection could lead to chronic rejection because some of these factors that are renal protective may be in fact. In fact, we also studied USAG1, uterine sensitization associated gene 1, you talked nicely about, and that's a very good marker for tubular cell integrity. We are also studying with NKCC2, and I did take down a few additional genes you talked about for future studies. We also found, this is for rejection, we also found that individual patients during rejection, some of them express a transcription factor called FOXP3. This is absolutely a fascinating factor. Again, all of you have this factor in your cells. If you didn't have it, you will have an autoimmune disease. This is absolutely essential for normal cell. Within our cells, we have T cells that can damage your cell. We have what are known as suppressor cells that prevent the damage, and this FOXP3 gene is very important. And we found that this gene could allow us to individualize therapy. We talked about not having population statistics, but individualized patient care. It's our hope that using these type of strategies by monitoring our urinary cell mRNA profiling, we will know the immune status of the patient, just like a blood pressure in a hypertensive or a blood glucose in a diabetic. We will know the immune status of that individual and fine-tune immunosuppressive therapy. This is our ultimate goal, transplantation tolerance, defined as transplantation without the requirement for long-term immunosuppression. In the laboratory, many of us can accomplish this, and we all have mice running around in our laboratory with islets or other organs. I'm very happy, no immunosuppressive therapy, but in the clinic, it has been very difficult to accomplish. And just in 2008, a landmark paper was published by a research group led by David Sachs. For the first time in the history of transplantation, the first time in the 50-year history of transplantation, HLA mismatched kidneys could be transplanted with patients without immunosuppressive therapy. And this is actually a product of many, many years by the MGH and the Harvard group. We participated in the study in a number of ways, and one of them was to do gene expression profiling in this patient and show that the FOXP3 cells are increased in this patient. And in fact, in the next 3 to 6 months, we will be initiating a very similar protocol at the center because NIH has expanded this study to include Penn and our group. And it's going to be a very exciting development. But we don't have enough organs. As today during the symposium, at least about 8 to 10 patients perhaps will die because of a lack of organs. We have 80 to 90,000 patients waiting for a kidney transplant, and each year we have about 10 to 12,000. So one of the approaches is to use organs from non-human sources, or a xenotransplantation. And we have to really modify. The pig is the most popular donor at this time. But there are to be many modifications that needs to be done, genetically modification. One, we all have an antibody called anti-gal antibody. So this gene has to be knocked out in the pigs, and that has already been accomplished. Our complement is very toxic to the pig, so you got to put in, by what is the technology known as knock-in, you got to put in a variety of complement regulatory proteins. And you also have to provide immunosuppression. And the other possibility, what one would call a stealth approach is a physical isolation. and Rogosin as a very robust program encapsulating islets. And I actually see this not as an either-or approach, but actually a combination of approach, because this is one of the most major challenges. There have been many disappointments along the way, but it is, I think, it is possible. And the final thing when you do xenotransplant, especially a pig xenotransplant, is always the concern about infection. The infection, one way you can prevent the outside infection from coming in by doing it in a proper facility, but unfortunately the pig genome has inherited certain viruses within itself. One of them well known is the porcine endogenous retrovirus, and there is a new wonderful technology called RNA interference or hairpin RNA, which could be used to knock down. So by knocking out certain genes and knocking in certain genes, you probably could modulate a pig to be a reasonable donor. What one would call a humanized pig as a counterbalance to what some people think some people behave like pigs. So this is a meeting about the future. This is a meeting about where we are going to be. I thought this would be an appropriate slide to finish with. Barry, I don't think you need to get up to stop me. I'm done. This is my last slide. Thank you so much.